Dear friends and my dear students, you all are welcome in the biochemistry classes. Myself, Dr. P.K. Verma, and today I shall discuss carbohydrates, their structure, chemical nature, and classification. The carbohydrates are very common component of our food. We take carbohydrates in rice, potato, corn, as well as in the form of sugar. But where these are produced and what is the basic form of their production? The carbohydrates are originally produced by blue-green algae and plants by a process called photosynthesis. Glucose is the main product of photosynthesis, but later on it is converted into other forms and from plants transferred to bacteria, fungi, and animals with food. When we use the name of carbohydrate, we use the suffix "ose", such as fructose, glucose, mannose, lactose, etc. Now, in a very elemental form, we can say that the carbohydrates are compounds made of carbon, hydrogen and oxygen, and the ratio of hydrogen and oxygen is 2:1 as it is in water. If a carbohydrate is heated in absence of oxygen or in absence of air, it gives out water and sugar charcoal is left behind. That means water was the component with carbon, and because of this the term carbohydrate has been given. That means hydrated carbon or carbon mixed with water. In this view, the empirical formula is CX H2Y OY or CX H2O whole into Y. However, on the basis of the ratio of constituent elements, carbohydrates cannot be defined because empirical formula is only suggestive, not confirmatory of any group of the compounds. In organic chemistry, there is a special property known as isomerism, and because of isomerism, two different compounds may have similar empirical formula. For example, formaldehyde HCHO is CH2O. Similarly, acetic acid CH3COOH or C2H4O2. These have the empirical formula CXH2YOY. But these are not carbohydrates. Now, lactic acid, dihydroxyacetone, and glyceraldehyde, these are three compounds with same empirical formula C3H6O3. Here lactic acid and dihydroxyacetone cannot be regarded as carbohydrate in true sense. Many people believe that dihydroxyacetone also is a carbohydrate. It is a triose, but in fact it is an isomer of triose. It is not truly a carbohydrate. So, we must have some reliable definition of the organic nature of carbohydrates. Sometimes a carbohydrate has less number of oxygen. For example, deoxyribose has one less number of oxygen. Fucose and rhamnose also have one less number of oxygen. These are called deoxy sugars. In spite of having one less number of oxygen, no chemical nature is affected. There is no chemical method by which carbon can react with water to form carbohydrate. Even it is baseless to consider them polymer of formaldehyde. So let us discuss what is the modern chemical definition for carbohydrates. Carbohydrates are polyhydroxyaldehydes or polyhydroxyketones in which there is at least one asymmetrical or chiral carbon that bears alcohol or a sugar group CHOH. Now this definition includes four essential properties to become a carbohydrate. Mind it, it is polyhydroxyaldehyde or polyhydroxyketone, has asymmetrical carbon bearing sugar group, that is CHOH. So the properties are, these are either aldehyde or ketones. There should be at least one asymmetrical carbon or chiral carbon. The third property is the asymmetrical carbon must bear an alcohol group, and the fourth one is there should be two or more alcohol groups. So if any compound has only one, two or three properties, not all the four properties, it will be not regarded as a carbohydrate. Chemically, the sugar group can react to show properties like carbohydrate, but it is not truly a carbohydrate. Here is the nature of asymmetric group. Carbon has 4 bonds and all the 4 bonds are involved by 4 different groups R1, R2, R3, R4, etc. In that case, the carbon is called asymmetric or the chiral carbon. Now here is the structure of lactic acid. It has a carboxylic acid group, but there is no aldehyde or no ketone group, so it should not be a carbohydrate. However, it has an asymmetric carbon number 2 having an alcohol also. So lactic acid gives some reactions similar to carbohydrate. The second compound is dihydroxyacetone. It has a ketone group. Carbon number 2 bears a carbonyl group. There are 2 hydroxyl also on carbon number 1 and 3. So alcohol groups are 2, but neither carbon is asymmetric and therefore it is not truly a carbohydrate. The third compound is glyceraldehyde. Now see, it has an aldehyde group. The second carbon is chiral bearing alcohol, and fourth carbon also bears an alcohol group. So there are 2 alcohol groups. one asymmetric carbon bearing alcohol group. Aldehyde is also its group property, and in this way it bears all the four properties of the carbohydrate. So glyceraldehyde is the true carbohydrate. It is a smallest aldose, but for becoming a smallest ketone, there should be one more carbon atom. If there are four carbon atoms, a ketone can form a carbohydrate. Dihydroxyacetone is an isomer of glyceraldehyde. It has no chiral carbon, so strictly it is not a carbohydrate. However, in cells, it readily converts into glyceraldehyde by an isomerase enzyme. For smallest ketose, the number of carbon must be 4. You can see here, these are the 4 carbohydrates. The first two are aldotetroases and the third one is ketotetroase. In ketotetroase, carbon number 3 is asymmetric and also bears sugar group or alcohol. It has 3 alcohol groups at carbon number 1, 3 and 4. This smallest form of ketotetroase is called erythrulose. Just take care of the name. When it is an aldotetroase, it is erythrose. But when it is a ketotetroase, it is erythrulose. Erythrose and erythrulose. The two pattern of writing the name is different in case of aldoses and ketoses. Now come to the classification of carbohydrates. Carbohydrates have been classified initially on the basis of the number of units produced by dilute hydrochloric acid hydrolysis or enzymatic hydrolysis. On this basis, carbohydrates have 4 classes. These are monosaccharides, disaccharides, oligosaccharides and polysaccharides. Earlier, disaccharide was included in the oligosaccharide and only 3 classes monosaccharide, oligosaccharide and polysaccharides were made. Now monosaccharides are the smallest unit of carbohydrate that cannot be further hydrolyzed into 2 smaller units. So after final digestion, all carbohydrate converts into monosaccharides. When a monosaccharide is made of aldehyde, it is called aldose. When it is made of ketone, it is called ketose. Here the ose suffix has been added to aldehyde and ketone groups. Similarly, on the basis of number of carbon, if there are 3 carbon atoms, the name is triose, 4 carbon atom tetrose, 5 carbon atom pentose and 6 carbon atom hexose. Higher forms are naturally not stable. There is a single compound pseudoheptulose that contains 7 carbon, which is formed as a temporary intermediate at the time of photosynthesis. But it readily breaks into triose and tetrose. Now, monosaccharides show optical isomerism. In the classes of chemistry, you might have studied stereoisomerism and the optical isomerism. If a compound contains at least one asymmetric carbon or chiral carbon, it has optical activity. That means it can change the polarity of light towards right or towards the left. If the number of asymmetrical carbon is n, the number of isomers will be 2 to the power n. For aldose it is n C minus 2 and for ketose n is C minus 3. Here aldotriose has two forms, aldotetrose 4, aldopentose 8, aldohexose 16 forms. But there is no ketotriose because it has no asymmetrical carbon. In the ketotetrose there is one asymmetric carbon, ketopentose has two asymmetric carbon, ketohexose has three asymmetric carbon, and therefore the tetroses has two types, pentoses have four types, hexoses have 8 types. In totality, triose has only 2 types, but tetrose have 6 types, 4 aldoses and 2 ketoses. Pentoses have 12 types, 8 aldoses, 4 ketoses, and hexoses have 24 types, 16 aldoses and 8 ketoses. Glyceraldehyde has only one chiral carbon, so it is either dextrorotatory D form or levorotatory L form. But from each of them, a family of higher compounds is formed by addition of new carbon atoms. In this way, compounds of D family and L family are formed. While forming higher compounds, the configuration of parent compound is maintained. Here you can see that in left side levorotatory glyceraldehyde is present and in right side the dextrorotatory glyceraldehyde is present. So from L-glyceraldehyde, 2 members of L-tetroses and from D-glyceraldehyde, 2 members of D-tetroses have been formed. Later on from each of the 4, 2 other members will be formed. In this way, by addition of carbon, one can make a family of higher compound from every isomer of glyceraldehyde. Here is the example of ketoses, erythrose. There is D-erythrose as mother compound in this example. 2 forms, D-ribulose and D-xylulose have been formed, and from each 2 other members of D families are formed. So from dextrorotatory a family of D family, from levorotatory a family or L family is made. Optical properties of these higher forms depend on the net effect of the optical properties of all such carbon. If the actual rotation is rightward, it is called plus form, and if the actual rotation is leftward, it is called minus form. Thus D plus, D minus, L plus, L minus are different groups of optical isomers. One cannot decide just by seeing a structure of any compound where it is D or L, where it is plus or minus. In fact, the structure is made after reading the optical properties. So, if the actual property of the mother compound is dextrorotatory, OH is placed in the right side. If the actual property is levorotatory, OH is made at the left side. In this way, the D and L families are made, but the plus or minus is difficult to decide in which direction these are placed. Every compound of L family has an opposite or mirror image in the D family. These two mirror images are called enantiomer, and all those compounds which are not enantiomers are called diastereomers. So except the two compounds, all are diastereomers. Similarly, if two compounds are compared and there is a single difference in the position of hydroxyl, these two compounds are called epimers. But if there are more than one difference, two or three differences, these are not epimers of one another. The number of epimer is same as the number of asymmetric carbon. So D-glucose would have three epimers, D-mannose, D-galactose and D-idose. In L family, the L-glucose also may be regarded as epimer. But mannose, galactose and idose are not epimers of each other because more than one difference, in fact two differences are there in each of them. In nature, ribose, deoxyribose, glucose, galactose and fructose are commonly found. D-ribose, D-deoxyribose, D-glucose, D-galactose and L-fructose are more common optical isomers. Now, Fisher and Haworth studied the structure of pentoses and hexoses. They found that the two types of structures are formed in them. These are furan and pyran forms. When 4 carbon and 1 oxygen atom form the ring, it is called furan, and when 5 carbon and 1 oxygen atom forms the ring, it is called pyran. The pyran and furan rings are formed by formation of hemiacetal between carbonyl of aldehyde and alcohol groups. The hemiacetal is formed between carbonyl and the 4th carbon in the furan and 5th carbon in pyran. The ketal is formed between carbon number 2 and the 5th carbon in the hexose sugars. The carbon of this aldehyde group C1 or ketone group C2 becomes chiral without affecting optical property when the ring structure is formed. This carbon is called anomeric carbon, and there are two possible forms of arrangement. Alpha anomer and beta anomers can be formed from them. This bond is formed reversibly. As a result, the chain can open and form closed ring in solution. There is always equilibrium between the straight chain and the closed ring structures. Now see how alcohol and aldehyde form the hemiacetal. Alcohol and ketone form the hemiketal rings. Here is a molecule of glucose. Hydrogen from the carbon number 5 moves to first carbon. Therefore, an alcohol is produced on carbon number 1 and a hemiacetal bond is formed. The structure of glucose may be like this, but for simplicity, a perfect ring is formed. Here is a fructofuranose. The hydrogen shifts from carbon number 5 to carbon number 2, and therefore fructofuranose beta type or alpha type is formed. In case of beta, the hydroxyl on carbon number 2 is below the plane. In case of alpha, the hydroxyl group is above the plane. In fructose, mostly the beta position is common. The ribose of RNA and deoxyribose of DNA are beta anomers. Glucose exists in both the forms, but beta anomer is more stable than alpha anomer. In plants, beta anomer is utilized in synthesis of cellulose, whereas alpha anomer is utilized in synthesis of starch, and therefore in our food, starch and glycogen are formed from alpha D glucose. Now, when two monosaccharide units join together, a glycoside bond is formed in between them and a disaccharide is formed. Maltose is the most common disaccharide. It is produced by hydrolysis of starch and glycogen by the action of alpha amylase enzyme. The milk sugar lactose has galactose and glucose, and the common sugar or cane sugar sucrose or invertose has glucose and fructose, D-glucose and L-fructose. Trehalose, commonly found in insects and arthropods, two molecules of galactose join together. In maltose and lactose, the covalent bonds develop between alpha position of carbon number 1 of the first unit and carbon number 4 of the second unit. So it is named as alpha 1,4 glycoside bond as shown here. In sucrose or invertose, alpha 1,2 glycoside bond is formed and the aldehyde group becomes locked, and therefore in neither unit there is reducing property. So sucrose becomes a non-reducing sugar, whereas maltose and lactose had second unit reducing in properties. Now, all monosaccharides and disaccharides can dissolve in water, and these are sweet in taste. So these are called sugars. But the oligosaccharides and polysaccharides are tasteless. There are some non-carbohydrate compounds which are sweet in taste, but these are not regarded as sugar, so these are called non-sugars. For example, you can see here the properties of some of the sweet compounds. The true sugar, fructose, is the sweetest sugar. 170 unit sweetness is there. The common sucrose is regarded as 100% sweet. Maltose is less sweet and lactose is very less sweet. On the other hand, synthetic compounds like aspartame is 160 times more sweet than common sugar. Saccharin is 400 times sweeter than sugar. And monilin is 2000 times sweeter than the sucrose. So monilin is the sweetest compound among the non-carbohydrate compounds. Now when monosaccharides and disaccharides, particularly the aldehydes or the aldoses, are tested for their reducing property, they give positive result. So these are called reducing sugars. Ketoses have poor reducing properties. Among disaccharides, maltose and lactose are reducing, but sucrose, the common sugar, and trehalose are non-reducing sugars. Oligosaccharides contain more than 2 but up to 10 monosaccharide units. These are smaller chains of monosaccharides. These are feebly soluble in cold water, are tasteless and non-reducing. They have no independent biological activity or the use, but these are formed at the time of hydrolysis of polysaccharides or at the time of their synthesis. However, as part of protein, they help in activities of hormones, enzymes, antibodies, and receptors. When these oligosaccharides become attached to a protein, they form the glycoproteins. Now you can see that a long chain of glucose can be made by formation of alpha-1,4 glycoside linkage. There are polysaccharides having large number of monosaccharide units, technically more than 10 units, but normally hundreds of units are there. These are formed in nature either to store glucose or fructose in the insoluble form for future use as source of energy, or for the formation of structural substances such as the cell wall of plants. Some of the common polysaccharides are starch. It is formed in green plants as polymer of alpha-D-glucose. There are two forms of starch. When it has only a straight chain made by formation of 1,4 glycoside bonds, it is amylose. But when branches are formed by formation of alpha-1,6 glycoside linkage, after every 20 or 22 glucose units, amylopectin is formed. This is the branching point where alpha-1,6 glycoside bond is formed, and in the straight chains of each of them, alpha-1,4 glycoside bond is present. So amylopectin has both alpha-1,4 and alpha-1,6 glycoside bonds. In the animals, glycogen is formed in liver and muscle. These are like amylopectin, but the branches are formed only after 8 or 10 glucose units. So these are more complex, more branched structures. A third very important compound is inulin. It is polymer of fructose found in the root tubers of dahlia, Jerusalem artichoke, and sugar beet, etc. Cellulose is made of beta-D-glucose by formation of beta-1,4 glycoside bond. Chitin is derived from galactosamine, that is amine derivative of galactose. They form an insoluble polymer and it is chemically very resistant. It cannot be digested easily by any animal's enzyme. However, bacteria can digest them. In addition to these forms of carbohydrates, there are many minor derivatives also, such as heparan sulfate found in connective tissues. Now, how the carbohydrates can be tested? The most common test is called Mollis test. If you have a soluble form of carbohydrate, dissolve it. Now 1 ml, 0.1% alpha naphthol is taken. 1 ml of sugar solution is taken above the alpha naphthol solution. If a drop of concentrated H2SO4 is put through the valve, when it comes near the interface of sugar and alpha naphthol solution, a purple violet ring is formed. If you will mix the reagent with the two solutions, the color will be pink. If you have to test for a starch can be dissolved in the boiling water. When a drop of Lugol solution, that is iodine reagent, is mixed in it, blue color appears. Glycogen gives very poor reaction with the iodine reagent. For test of reducing sugars, Benedict's reagent, Barfoed's reagent, Fehling solution, etc. can be used. Benedict test is most common, in which on heating mixture of sugar and reagent gently, the color changes from blue to green to yellow to orange and finally to brick red. So depending on the concentration of sugar, there is different types of coloration in the mixture. In addition to these tests, there are anthron reagent for glucose test, resorcinol for fructose, and orcinol for ribose sugar, etc. These reagents also can be used for quantitative estimation of glucose, fructose, or the ribose sugars. So you have learnt much about carbohydrates. In the next session, we shall discuss another topic such as lipid. Thank you very much.